Great. Thank you very much. And thanks for the invitation to be here today. Many of the things that you're speaking about today are paralleled in our field in glaucoma. You could substitute the word glaucoma for diabetic retinal disease any place along the conversations thus far, because we're grappling with the same issues that you are. For glaucoma neuroprotection clinical trials, these are some of the issues that we face, and you could just change the title for yourselves, of course. And today we're talking mostly about endpoint selection, and the regulatory and registration requirements that might eventually be required. And this, of course, relates not just to phase 3 that a lot of people look towards, but also for phase 2, which for us is probably more important in terms of developing new products, new strategies, new techniques. What are the tools that we have in glaucoma? They're very similar to the tools that you have. We have data from longitudinal studies, and there's some regulatory guidance out there. This is just a sample of some of the glaucoma trials that we have. You're all familiar with some of these names: the ocular hypertension treatment study, following patients with 20 years, randomized to control group, untreated, versus treatment of elevated intraocular pressure, and so on. The last two on this list were attempts to look at neuroprotection in clinical practice, for which we learned a great deal. Here's the most recent pressure-lowering randomized trial, the United Kingdom Glaucoma Treatment Study. There are 250 patients roughly in each arm, followed for many years, but there was a separation in treatment effect at 2 years with intensive visual field testing. You heard before from Ted, you need lots and lots of visual fields over time to detect change. This has been one of the problems for us. And these patients had 2 visual fields roughly every 3 months. And you could just see the separation between the groups even as early as 1 year. It's a lot of patients. But on the other end, looking at neuroprotection, pressure-independent change in glaucoma patients, with a relatively small number of patients, about 125 in this entire study, we could demonstrate a difference between 2 accepted treatments, one that has a putative neuroprotection effect in patients. And even if that's not the actual mechanism here, at least we could demonstrate separation between the 2 groups with relatively fewer numbers of patients. There is, of course, regulatory guidance here for a variety of eye conditions, dry eye, inherited retinal diseases, and so on, but there's really nothing for glaucoma and neuroprotection. And so we attempted to solve it the same way you're trying to solve it today, by getting everyone in the room together and work together and create some common ground for the future discussion. These were 2 published articles led by Bob Weinreb and Paul Kaufman that brought together the FDA, the NIH, ARVO, the entire glaucoma community to look at possible strategies. Most recently, you may be familiar with the neuro-ophthalmology meeting that was held. I just highlighted a couple of things here because the neuro-ophthalmologists consider glaucoma under their domain. We'd like to hand it off to the retina specialist, by the way. The ganglion cells are in the retina. You can have it. But what it really taught us, and what we learned from the first 2 meetings, was that regulators in particular are very flexible, provided we provide them with the right information. Here you see some interesting information, caveat that the results of automated perimetry with clustering. Clustering the visual fields is a relatively new concept, doing maybe 4 visual fields at baseline and a couple of months later doing another 4, just in a very short-term trial, to try to see if there's any look for treatment effects. There's also no consensus on structural endpoints, and you can read that right there. That there's no real, in glaucoma at least, we've been yet able to demonstrate the correlation between OCT structural change, for example, and visual field change down the road. Or I think we're pretty close on that. What do we have? We do have some prior studies that had accepted endpoints for phase 3. The first one here is from the memantine trial, which I'll speak about in just a moment. I'm looking at where progression was defined as 5 or more reproducible points using a glaucoma change probability analysis, and there are others. You'll note that it says structural endpoint. That would be a non-reversible OCT or neurofiber layer change measured by OCT over a 3-year period might be acceptable, but the amount of change that's clinically relevant has yet to be defined. And that's up to us to define. People in this room need to define what that means for diabetes. We also know that a quality of life endpoint at the moment is not acceptable from the glaucoma perspective. And durations become a huge problem. Duration of a trial, the memantine trial was 4 years in duration. And of course, there's safety data that has to be collected. So what's our biggest hurdle? Our biggest hurdle is that we can't proceed to a reasonable phase 3 without a phase 2 program. And I'm pretty confident we can design a phase 3 program that would be acceptable to regulators, to industry, and so on, but we have no viable phase 2 programs. Because of that, the memantine trial was a combined phase 2, phase 3. It was a huge gamble, 2,300 patients, worldwide project. The outcome measure I described to you a little bit earlier is highlighted in red. And this trial failed. It cost lots of money. And we learned a lot, that the FDA was flexible, but there were lots of hurdles. And this just speaks to that in the inherited retinal disease guidance, that there is a lot of flexibility. To try to address that, we recently performed a study looking at nicotinamide and pyruvate for glaucoma using that clustered approach. Many of you are familiar with the basic science that came out of Simon John's lab. It's an excellent article in Science, where he demonstrated that B3 could be neuroprotective in a mouse model of glaucoma. This is our study design. It was randomized, it was prospective. And you can find all these, the slides are actually available on the JAMA Ophthalmology website. Essentially, all we had were 32 patients. COVID interrupted us a little bit. The target number was about 36. But over time, in a 2-month timeline with clustered visual fields, 4 in the beginning, 4 at roughly 2 months, we were able to generate an uptick in sensitivity in a number of points in the field, demonstrating there was some improvement, meaning that the drug actually got to the target tissue, had some effect at that target tissue using this novel strategy. That was also reproduced using the phototopic negative response by Johnny Croston's group in Australia, describing the same thing. And because there were 2 separate phase 2 type of trials that were successful, there's now a worldwide effort to look at nicotinamide in glaucoma in a phase 3 equivalent type of trial. This is just our study design with about 200 patients with intensive visual field testing over 18 months. And we learned a lot from these different short-term trials. And because of that, as I said, there are 5 programs worldwide now that people are committed to performing. Of course, there's no pharmaceutical support because they're nutritional supplements, but there are governments that are willing to support them. Just like to bring this to your attention when we talk about moonshots. We live in an age of wonderful science, and because of the vaccine, development of the vaccine in a year for COVID, the entire world, the business community, is expecting great things of us. We have wonderful technology. I think we're capable of applying it. We need to get out of the box a little bit, which you're trying to do here today. And the glaucoma community would like to come along for the ride. Thank you.
Thank you very much. We should have some questions here.
One closet glaucoma person.
I was surprised that you said that there is no correlation between anatomy and function when there is so much papers, maybe not as important as they should, showing that there is correlation between different measurements on anatomy and function, and how we can end up with this.
So there's a huge trove of scientific literature that correlates structural appearance as measured by OCT, let's say, and visual function. The question is, how does one link the structural change to a clinically significant change in function that would be acceptable to a regulator who needs to approve the drug? That is the linkage that is missing. I won't say that it's not possible. It is possible. I think that we need to create, through meetings like this, define that linkage, and then be able to move on into the regulatory realm. And as scientists, clinician scientists, we're satisfied with that linkage. We know that the structure and the function go along together. Actually, the structural measure with OCT is clearly more sensitive to change than the functional measure. It's more reproducible, it's more reliable, more repeatable. detect the change earlier, smaller amounts of change. But a tiny little change, a 1 micron change in OCT thickness, is that meaningful for a patient? I can't answer that question. Right? So I think we need to.
Thank you, Jeff. Wonderful talk. I wonder it's on. So you mentioned that your phase 3 trial, I appreciate that it's not a regulatory trial in the sense of a drug discovery. I wonder whether that, how would you think, or some other people in the audience, I don't want to put Dr. Chamber on the spot, would that be acceptable if you were a new drug? The question is like, well, would that be clinical meaningful, and what will be your primary endpoint?
So the regulators, I don't want to speak for the FDA, but the regulators have given us some guidance. And there's 2 thoughts related to that. One is to propose new endpoints, discuss with the agency with data, and develop a new endpoint. It could be contrast sensitivity, could be something else that could be validated, or take a look at the endpoints that currently exist and see if the data that we can collect in a 2 to 3 year period actually meets those endpoints. The way the endpoints were described, by my understanding, is that you need a certain number of points in a certain location in the field to change by a certain amount. Let's just say it's 7 dB, for example. When we measure mean deviation in the field, and the field is progressing very slowly, the entire field, that doesn't mean that the points are progressing slowly. That means most of the points are not changing, but within the field, there actually are points that are progressing quite rapidly, even over the 2 year period. So it's quite possible that the description of the endpoints that currently exist, ones I had on the screen, are achievable in a relatively short period of time. For example, at the edge of an existing scotoma or the edge of disease, of visual field deficit. And we're not too far off. We're not too far apart. So actually, one of the reasons we want to do the trial, a longer term trial, is to answer that question precisely. We kind of know what the effect of the B3 might be, but it's an exercise for a new drug. We'll know whether we can actually meet the endpoints that have been previously described in a short period of time, or whether we need to tweak that, have further discussions with regulators around the world to see what else we can use. But there are a lot of different possibilities here. In glaucoma, we tended to think about peripheral loss, but the central loss is much more important than peripheral loss to patients. And perhaps there are endpoints that we could agree upon that are related to central visual field damage, which would relate to this group as well.
Thank you very much, Jeff. That was fantastic. Right on target.